Hi, this is Anna Master. Welcome to our synchronized session, and tonight we're going to talk about genetics and genomics briefly. So when we're talking about genetics and genomics, we're talking about the DNA. And in our cells, particularly in our nuclei, our DNA is housed, and it is the manual for our genome. Okay, genome, genes, DNA, referring to the same thing. And this molecule of our genome is written in DNA molecules, so it's written as A, C, T's, and G's. So instead of being binary, like a computer, where we're either on or off, we're quaternary, as in DNA. Okay, here we go. All right, so what exactly is a gene? A gene is a sequence of DNA that is encoded for a particular protein. Genes can have different forms and can vary from individual to individual. This is referred to as an allele. For example, genes for blue eyes versus genes for green eyes. Genetics is the study of inheritance. And a genome is your complete set of genetic instructions. Now, there's another term called exome, and exome refers to the protein coding genes, and it's less than 2% of our DNA. So, in other words, the human genome consists of about 40,000 genes. The rest, the other 98% of our genome, we don't exactly know what it does. And environment influences how genes are expressed. For example, if you have the genes for freckles, if you stay in the sun, if you're, like to surf or on the beach all the time, you'll probably have more freckles. Whereas if you have the same gene, an identical twin who has the same gene for freckles but is a bookworm and stays in the library, they may not have as many freckles. Okay. And what this is showing you, again, in the nucleus of the cell, you have your chromosomes. Chromosomes are made of DNA, and you have 23 pairs of chromosomes. But only a small part of the DNA, this part right here, is a gene, is going to be made into a protein. And remember, your proteins are your cellular machinery. They're your enzymes and hormones and receptors and muscle and hair. There are many, many things. Okay, so now we're going to talk briefly about modes of inheritance. And what genetics can do is predict. And the reason why we can predict is we have to know two things. We have to know meiosis and how your genes are going to be distributed during meiosis and what combinations will occur or could possibly occur should fertilization take place. So you can calculate the probability of certain traits that will appear in the offspring of two individuals. And these patterns in which the genes are transmitted in families is often termed mode of inheritance. Okay. So as I said earlier, you have 23 pairs of chromosomes, and they're all laid out right here. The first 22 pairs are called autosomes. In other words, auto meaning the body, they determine, height and eye color and things like that. Pair 23 is called your sex chromosomes, and they determine your gender. I guess actually a better term for that, they determine your sex or what gonads you have. A gene, again, is nucleotides. Again, nucleotides are the building blocks of DNA, and they can they can exist in various forms called alleles. Again, the difference in allele could be the gene for blue eyes versus the gene for green eyes, what type of blood you have. You could have A or B or O. These are all determined by alleles. They're coding for the same protein, just a different form. If you have two identical alleles, you're called homozygous. Homo meaning same, zygos meaning referring to the fertilized egg. And if you have two different alleles, you are heterozygous. So what alleles you have is called your genotype. And what you see is the phenotype. So you may get the gene for, let's say, brown eyes, and one gene for brown eyes and one gene for blue. However, your phenotype will be brown. Wild-type alleles produce mutations. Normally, wild-type is the most prevalent allele. Mutation is just a change in the sequence. So it's what produced the different eye colors and the different hair colors and things like that. Okay, so when someone is heterozygous, one allele may determine the phenotype, and that would be the dominant allele. So when I gave the example of someone having blue eyes, or having the gene for one gene for blue and one gene for brown, brown would be the dominant allele, because then it would mask the phenotype of the recessive. You would be unable to tell that that person had blue eyes, or the gene for blue eyes. Recessive can only be expressed in a double dose. In other words, eye color is not the best example here. So say, for example, if you're a carrier for cystic fibrosis, you need to have two genes in order to have the illness associated with cystic fibrosis. You have to be homozygous. Autosomal conditions are carried on the non-sex chromosomes, chromosome pairs 1 through 22. Sex-linked are carried on chromosome 23. And we'll talk about those in a minute. X-linked conditions are carried on the X chromosome. X-linked conditions are usually passed on from mother to son. And then Y-linked conditions are carried on the Y chromosome. And anything Y-linked is transmitted from father to son. And we'll talk about that a bit more in a moment. Okay. So basically what we're doing here is a Punnett square. We're taking two parents here. One is normal and the other both parents are normal. That's their phenotype. But their genotype, they are carriers, meaning they have one wild type and one gene for cystic fibrosis. So when meiosis occurs and you predict the possible genomes that could arise from mating there, you get three possible outcomes. One outcome is that the child will receive both normal genes, each normal gene from the parent. So they will have the genotype. This is their genotype is normal, and their phenotype is non-cystic fibrosis. Two will have the genotype having one cystic fibrosis gene and one normal gene. Both of these, okay. Their phenotype will be normal. Now one child, or there's a 25% chance that a child will get both pairs of cystic fibrosis gene. This is a genotype, and therefore the phenotype would be a carrier of cystic fibrosis. Now the thing to remember with Punnett squares, which is what this is that this is not predicting what's going to happen should they have 4 children. So should they have 4 children, 3 will be normal and 1 with cystic fibrosis. This is telling you the percentage of passing on cystic fibrosis every time they have a child. So every time they have a child, that child has a 75% chance of having the normal phenotype, and a 25% chance of having cystic fibrosis. Now, that is recessive inheritance. In other words, cystic fibrosis can be masked by the wild-type gene. Here we're looking at a dominant inheritance, and for it to be dominant, you only need one copy of that gene. And an example of this is HD, or Huntington's disease. The reason why this gene usually gets passed on is because the symptoms of Huntington's disease usually don't appear until the person is in their late 30s, early 40s. So many people have already had their children prior to knowing they have this allele. So if one person is a carrier and thus affected, and the other person is not, their children are divided into, there's two possibilities. The genotype would be normal, and their phenotype would be unaffected. The other half would have one gene for Huntington's disease. The other is normal. And because this is a dominant gene, their phenotype would be that they would express the Huntington's disease. And of course, not everything works as completely dominant or completely recessive. There's a couple terms here: incomplete dominance and codominance. If you have incomplete dominance, that means a heterozygote has an intermediate phenotype. And this example here is hypercholesteremia. If you have wild type, you don't have it. If you have both genes for it, you have really high cholesterol levels. But if you have, if you're heterozygous, your cholesterol levels are in the middle. Codominance means that both alleles are equally expressed. And the best example of this is a person with AB blood type. So they have the allele for type A and the allele for type B. So if you get both alleles, you have type AB blood. Again, type A and type B are dominant. So if you get one or both, you can have type A. If you get one B or two Bs, you're type B. Type O, you get neither gene or neither allele. So type O is recessive. And this brings up a good point. Just because something is recessive doesn't mean it doesn't occur much. Dominant and recessive have nothing to do with how often they appear in the population. Because in America, the majority of people are type O, even though it's a recessive trait. Dominant just means it can mask a recessive trait. And let me see here. Now, most genotypes can vary from person to person. And these are what we're referring to as penetrance, expressivity, and pleiotropy. And so these are the distinctions. Complete penetrance means that everyone who inherits this allele has some symptoms. Incomplete penetrance means that only some individuals will express this phenotype, even though they inherit the alleles. An example of that is polydactylism, which is an extra digit. So in other words, it's variable expression. If you are polydactyly, you may have a complete extra digit, or you may just have a little bit of a digit. Pleiotropy is referring to a genetic disorder producing several symptoms. An example of this is Marfan syndrome. And what Marfan syndrome is a mutation in a connective tissue, connective tissue fiber. And because of that, people develop a wide variety of symptoms. So this one gene affects many phenotypes. Genetic heterogeneity. Now this is hetero meaning differently. That means the same phenotype is the result of the actions of many different genes. Deafness is an example. There are many genes influencing. Many genotypes can cause the phenotype deafness. Dwarfism is another example. Many different genes can regulate the phenotype of dwarfism. Okay. Now another caveat in our genetics lecture. Most but not all characteristics are inherited. Some genes are what are called, or some traits are what are called multifactorial traits. And the traits are molded by multiple genes. This could be height, skin color, and many common diseases. So this is skin color. And there are 3 genes that determine, and it's therefore polygenic, poly meaning many. Okay. In matters of sex, which genitalia you have the human somatic cells, the non-sex cells, and then you have the X and Y chromosomes. Females have 2 X's, whereas males are X and Y. So all eggs can carry a single X chromosome. So a mother, her oocytes are always carrying the X chromosome, whereas the sperm can either be X or Y. In other words, it's the father of the child that determines the gender of the child or the sex of the child. Sex is determined at contraception. So a Y-bearing sperm fertilizes an egg and conceives a male, and an X-bearing sperm conceives a female. Sex determination. So maleness derives from the Y chromosome in the gene is called SRY. And this gene encodes for a protein, and this protein leads to the development, the SRY, or this protein is called a transcription factor, and this transcription of genes lead to the development of the male structures in the embryo. In other words, all babies, when they're forming in utero, the first part are female. But once the SRY gene kicks in, then those with a Y chromosome will form male characteristics. Another thing you might have noticed was how large the X chromosome is compared to the Y chromosome. So the X chromosome has over 1,500 genes, and the Y chromosome has only 231 genes. This is where the X-linked and Y-linked come in. The X-linked, most genes on the X chromosomes don't have corresponding alleles on the Y. So in other words, these genes are passed from mother to son. Now, of the 231 genes, there are, and some of these are unique to the Y chromosome, these are passed from father to son. An example of an X-linked disorder is hemophilia. And the son has a 50% chance of receiving the recessive allele from the mom. And And every time, because this recessive allele is on the X and the male only receives one X, one recessive allele will cause hemophilia. For a daughter, for female to have hemophilia, and females can have hemophilia, both of their X chromosomes need to have the recessive allele. Now, if only one of their X chromosomes has the recessive allele, they are what's called a carrier, and they can pass hemophilia on to their sons. Sometimes gender, let me find my cursor here. Sometimes gender affects phenotype. Some traits are what are called sex-limited. They affect structures or function in the body that are present only in males or females, such as beards and growth of breast. Some are sex-influenced inheritance. Can be dominant in one sex and recessive in another. Baldness. Heterozygous males are bald, but heterozygous females usually are not. There are many chromosomal disorders. In other words, you can have an excess or deficit of chromosomal disorders. Now, when you start messing with the number of chromosomes, and again, humans have 46 chromosomes, there are not too many disorders where the newborn child will come to term with extra or missing chromosomes. Polyploidy means they have, well, actually they have a drastic upset in chromosome number. So they have many more than 46 chromosomes. And it could be an entire set of chromosomes. And most embryos or fetuses die. But if those that do survive have a very short lifespan. Aneuploidy means you have missing or have an extra chromosome. And that results from a meiotic error called non-disjunction. Non-disjunction is when the homologous pairs fail to separate during meiosis, particularly meiosis 1. So this could result in a zygote either having 47 or 45 chromosomes instead of 46. Trisomy is having an extra chromosome. An example of that is Down syndrome, trisomy 21. Monosomy is missing a chromosome. And let's talk about some of these disorders. Trisomy, in the autosomal chromosomes, only 3 chromosomes can have trisomy with the and have the child born. 13, trisomy of 13 and 18, they are rare and survival is not very good. However, Down syndrome, which is trisomy 21, they can live a fairly long life. Let's see. Now, if you are missing a chromosome, for example, if you're missing one of your sex chromosomes, so you just have an X, that's referred to as Turner syndrome. Sometimes you can get 2 X's and a Y, that's Klinefelter's. Poly-X means you can have 3 X's or 4 X's. And then Jacob's syndrome is having 2 Y's. Now, to test chromosomal abnormalities, they can do a number of tests. They can do maternal serum markers. They can do amniocentesis, in which they remove part of the amniotic fluid and look at the karyotype. The karyotype is just a mapping of the chromosomes. CVS, or chorionic villi sampling, This is the villi that will make the placenta. This will lead to a karyotype. You can do fetal cell sorting. I'm not completely familiar with that one. And also, another one is ultrasound. Now, you're not looking at the genotype. You're looking more at the phenotype. But this can give a very nice test to see if there are any abnormalities. And I believe this is the end of the lecture, or end of my presentation. Thank you very much, and I hope to see you next semester.